Hey, welcome to Myelin. My name is Ryan Pevey. I have a PhD in neuroscience, and today I want to talk to you about neuronal evolution in the human brain and how that might be tied to autism. So today I'm going to cover this paper called "A General Principle of Neuronal Evolution Reveals a Human Accelerated Neuron Type Potentially Underlying the High Prevalence of Autism in Humans." And this is a really cool paper that was published a few months ago. I didn't catch it right away when it was first published, but I picked up on it pretty recently and got really excited about it when I saw it. So they started with the hypothesis that more abundant cell types were under a greater selective constraint and thus slower evolutionary divergence, I should say, than less abundant cell types. Because a change in gene expression in the more abundant cells would have an effect in a larger number or a larger proportion of cells in the organism. I think that's a really interesting hypothesis. It's the first time that I've come across this kind of hypothesis. I don't know if this is kind of like commonly accepted in the evolution field as looking at the evolution of specific cell types, but I think that's a really interesting hypothesis. And what they found in the first figure, they're kind of going through and they're establishing that they did in fact find this to be true, right? So they found the rule to be generally true across all three studies that they looked at. In three different brain regions. And really what they were doing is that they were taking data from three previously published single-cell RNA-seq papers that contained, really what they were looking for is that they were looking for data sets that contained human data and then several other mostly primate outgroups that they could look at. Right, so they're looking at neocortical samples in all three of these. So one of them was from the middle temporal gyrus. That included middle temporal gyrus tissue from humans, chimpanzees, gorillas, rhesus, macaques, and marmosets. The second data set that they looked at was looking at dorsolateral prefrontal cortex tissue, DLPFC tissue, from humans, chimpanzees, rhesus macaques, and marmosets. And then the last one that they looked at was primary motor cortex from humans, marmosets, and mice, right? So they were able to look at all three of these data sets in three different regions of the neocortex, and they could find that, generally speaking, this hypothesis held to be true, which is that the more abundant cell types were under greater selective constraint, and that essentially that those neuron populations would evolve at a slower rate over the course of evolution, right? So they're looking at all of these different species so they can see kind of comparing all of the different groups. The more closely related groups, evolutionarily speaking, would see a certain amount of divergence, and in the higher proportion neuronal populations, they would also see that there was kind of like a slower rate of divergence in those populations across all of the different species, right? So they found that rule to be generally true across all three studies in different brain regions in the neocortex. In their second figure, they also show that this relationship holds to be true if you look at just excitatory neurons or just inhibitory neurons, right? So this relationship appears to be pretty strong. So after that, they show that this relationship appears to be driven by genes that are either highly expressed in a given neuron type or that are very cell type specific, right? So they're kind of going through in the first couple of figures and they're showing that this relationship does appear to be true, and it seems to be true driven by specific subsets of genes within those specific neuron types. So that's all well and good. So after that, they established that this relationship in their first hypothesis does hold to be true, right? Where more common neuron types undergo less evolutionary change. It's a really cool result. I think it's super cool. This is a really cool type of analysis and a really good use of these single-cell RNA-seq data sets that are out there. So after they established that, they then further hypothesize after that any cell types that diverge from this rule might represent a population of cells that went through what they call atypical selective forces. So they look to see if there are any neuronal populations that diverge from this relationship in humans, specifically in humans. Basically, that means that in humans, these neurons evolved in a human-specific manner, and might offer some insight into what makes our brains special compared to other animals, right? So there's a lot to unpack in a statement like that. So let's talk about it for a second. First of all, everything that I'm going to say for the next couple of minutes is my opinion. I want to stress that this is me kind of like interpreting this paper and the results of this paper. This is not in the paper itself. It's not the opinions of the authors in the paper. But I feel confident that what I'm saying is more or less in line with scientific consensus. And these things are often just left unsaid in the scientific literature, because if you can't verify a statement, because it's either conjecture, which is kind of what I'm doing right now, or because it lacks hard evidence that's not being shown in that specific paper, then you leave it out of the primary literature. So I want to stress that what I'm about to say is really my interpretation and my kind of general personal thoughts about this, but generally speaking, it's widely regarded that a big part of what makes us humans special is our cognitive abilities. And there are a few reasons why our brains might be more capable than other species. First, the sheer number of neurons that we have in our neocortex is as much or more than any other species on the planet, right? Talking specifically about neurons in our neocortex, right? So if you include more than just neurons, or if you include the whole brain, things that are kind of like beneath at a lower level than the neocortex, then that doesn't necessarily hold to be true, especially when you start looking at the cerebellum. If you include the number of neurons in the cerebellum, then humans look a lot more typical, and generally speaking, larger animals like whales and elephants and other types of cetaceans like dolphins and stuff like that, they tend to have all of a sudden more neurons when you include the cerebellum, because a larger body typically means more neurons in the cerebellum, right? Furthermore, the neurons that are there are more branched and interconnected than in any other species in humans, right? So these two facts together mean that we have the most complex, most interconnected neocortex on the planet. And we also think that that is why we are also the smartest animal on the planet, right? So that was just kind of like my 2 cents on part of the background on why I think that this is like a really interesting study to do. So to answer the second hypothesis, where they're looking for neuronal populations that diverge from this relationship that they established in the first hypothesis of their paper, they're looking for evidence of faster divergence in specific neuron types in the human lineage compared to what their previous model that they set up predicts would be based on cell type abundance, right? To do this, they quantified the divergence observed in each cell type of the middle temporal gyrus data set in humans and chimpanzees using the gorilla samples as an outgroup. So again, in plain English, really what they're doing is that the difference between the gorilla and the chimp samples in that middle temporal gyrus data set can be attributed to divergences that happened in the Trump lineage. And the difference between gorilla and human samples can be attributed to the human lineage, right? So they're looking at basically the two different branches that you can kind of derive out of that data set. When they performed this analysis on the chimp data, they found that the neuron types diverged in line with their relative abundance, just as their model predicted, with a statistically significant negative Spearman's correlation coefficient of negative 0.77, right? So basically what that's saying is that when they look at the chimp data set, really it just kind of aligned with the expectations of the previous model that they put forth. However, when they looked at the human data, they found that there was not a statistically significant correlation, negative or otherwise. Now, you might think that this is a bad thing, but when you look at the graph, you see that there are a few populations of cells that stand out as not following this model in the human data. And the populations that they were most interested in would be anything that was high in cell abundance and high in evolutionary divergence, which corresponds to the top right corner of this graph. The cell population that is highest and to the right of this graph is labeled in their graph as the L23IT, or layer 2-3 intratelencephalic neurons. So what they did next is remove this population from the data set, rerun this correlation analysis. When they did that, boom, all of a sudden the data set shows a statistically significant negative correlation between cell type abundance and evolutionary divergence that they would have predicted with their previous model. And once they removed that cell type, they ended up with a Pearson's correlation coefficient of negative 0.59, right? So it's back in line with their model, right? So they looked at the human data, didn't line up with their previous model that they established, removed this one cell type, and all of a sudden all of the human data lines back up with the model that they suggested earlier, right? So that really lends itself to suggest that these layer 2-3 intratelencephalic neurons that they've isolated out of this analysis are special in humans in some way, right? They're part of that second hypothesis, where a population that diverges from the model from the first hypothesis might be special in some regard, right? So they found that these layer 2-3 intratelencephalic neurons appeared to be undergoing some type of selective pressure that was unique to the human lineages in the data set. Next, they wanted to look at any connection of phenotypes of disease in humans associated with these neurons. So they looked at two different databases of genes documented to be associated with some disease phenotype. They looked at the Human Phenotype Ontology, which is a repository that includes a broad swath of disease types covering more than 18,000 terms. So basically, this is a website that you can go to. You can go to this right now. You don't have to be a scientist. You can go to it. I put the link in the description, and either download the whole data set like the authors did, or you can do a search for basically the disease that you want and find a list of genes associated with that disease or condition, right? So you could look at cardiac arrest and then see what terms kind of show up along with that. You could look at diabetes and see what genes are associated with that in this data set, right? So that's kind of the data that this data set shows. The other database that they looked at is called Safari, called the Simons Foundation Autism Research Initiative. I'll put the link in the description for that one as well. And it does a similar thing. It's a similar type of database, but it's specific for autism spectrum disorder, right? So this might seem kind of random, but actually these layer 2-3 intratelencephalic neurons have been associated with autism in previous studies, like GWAS studies and other types of studies for a while. So this is not a surprise to me at all that they may have looked for a connection to autism spectrum disorder once they isolated these neuron types. And lo and behold, when they looked at the genes that are found in both the middle temporal gyrus data set and the dorsolateral prefrontal cortex data set, and they correlated those with the Human Phenotype Ontology data set and the Safari data set, they noticed that they didn't see a lot of correlation with, generally speaking, with the HPO, the Human Phenotype Ontology data set, but they did see that Safari dataset genes were selectively downregulated in these neurons, suggesting that there's some vulnerability in these neurons which uniquely undergo some type of human-specific evolutionary process, that those neurons are selectively vulnerable to autism spectrum disorder. So really cool result, really cool finding of this paper. That's kind of like the main tentpole finding of the entire paper. After that, they did some really important validation experiments where they took some human chimpanzee hybrid cortical organoid cell cultures, where they're taking cells that have been derived into neurons from stem cells from humans and chimpanzees, and they're combining them together to create these cortical organoid cell models that are really cool. And then they're looking at those to see whether or not those also correlate with the previous results of the rest of the paper. And they found, very importantly, that this data set also backs up where they see that there's some of the selective vulnerability that's found in the previous data sets also seems to be selectively vulnerable in the neurons in these mixed cell type cultures, right? So really cool study, really important validation experiment. Okay, so really cool study, really cool results, but what all does this mean, right? So interpreting this, right, this really shows, I think, that there is some kind of connection between the kinds of evolutionary changes that the human brain went through over the course of our human lineage, evolving from our previous common ancestor with chimpanzees. And a lot of those changes were really important to making humans humans, but it also shows that some of those changes resulted in a lot of the biggest strengths of humans and resulted in a lot of the really strong cognitive abilities that we seem to uniquely have as a species on this planet. But it also left us vulnerable to some of the conditions that we see out there, and autism spectrum disorder is one of those. And that's a big kind of open question in neuroscience, right? Is are humans actually vulnerable to these things? Or are we just not observing those things when they occur in other species? Or are humans truly vulnerable to these conditions in a unique manner? And I think this is part of a body of evidence that's really starting to show that autism spectrum disorder is a normal part of humans as a species, that humans in specific are particularly vulnerable to a condition that looks like autism spectrum disorder, whereas we're really just not going to see it in other species. They looked at other conditions as well. They also looked at schizophrenia, which is another condition that humans seem to be kind of uniquely vulnerable to. And they did see some results that are somewhat in line with us being uniquely vulnerable to schizophrenia as well. But they said that they kind of lacked the statistical power to be able to more conclusively say the same thing about schizophrenia, that there are specific neuron types, at least within this data set, that are vulnerable to schizophrenia. One of the things that's kind of missing from this in an evolutionary sense, of course, because we don't have the samples to be able to study them, is other human lineage species, right? So the closest species that we have to be able to study nowadays, of course, is chimpanzee. But they don't have, they don't have like Neanderthal, they don't have Denisovans, they don't have any of the other like human lineage species that are along the way, because of course we don't have any brain tissue to be able to do the single cell RNA sequencing sample on that. So you could, if you had, a Neanderthal available to you and you could get a brain tissue sample from them, then you could do this similar analysis and look at what are some of the brain type proportions that are present there, and whether or not these features are unique to like specifically just us Homo sapiens, or as part of a lot of the other human lineage species that occurred in the past that have now gone extinct. And of course, we don't know that, right? But there are some really interesting links in other parts of the literature that also kind of speak to the relationships between humans and Neanderthals and some of the other species that are out there as well. And we'll probably cover some of those on this channel here in the future. I think that's it for today. And I appreciate you watching, and stay tuned for other updates that are coming from the channel. I'm going to do these types of channels, talking head channels, where I'm talking to you specifically about news that's happening in neuroscience or other science avenues that I think are important or interesting that I want to cover here. But I will also probably do some updates with other papers that I think are interesting, just like this one. And I'm working on some more in-depth instructional videos that talk about some of the really interesting processes that happen on the cellular and molecular level within neuroscience and development and evolution of the brain. So keep an eye out for those things as well. I'm in the process of making some of those videos and editing those, and a lot of those involve some really cool 3D animations that I'm that I'm teaching myself really how to do, and has been a really fun and rewarding experience. So keep an eye out for those things as well. And, I appreciate you watching my first video and being here. Like and subscribe, I guess. I've never said that before. So welcome to my channel, and I hope to see you all very soon. All right, thanks. Bye. So they established this relationship in the first hypothesis, right? And I don't know that that's a great intro. Let me start all over again, okay? So in this paper, effect on a larger number of cells. What are they doing here, right? So let me think of the right way to say this. So they looked at furthermore, the neurons that are there, sorry, I'm dog sitting, where they're looking for those, they did find a slight. Now, one of the things that you you can stay there. All right. Getting photobombed by a dog. Cool.